powering a more recessive version of that. So he gave us the notion of a dominant trait versus a recessive trait. We did look at an exception when we look at incomplete dominance. That does appear to be a blending-like idea, so don't get it confused. It is kind of blending, but what it really means is that the dominant trait is working, but so is the recessive trait. They're both producing their proteins. So we see the effect of each. So it does kind of look like a blending issue.
Keep in mind that in biology, there's always going to be an exception to the rule. Nothing is ever black and white, except for our PowerPoint slide.
All right, now we want to get to a little more of a complicated one. It's really not complicated at all. It kind of depends upon, though, how you go about trying to do this. This is how I was taught in high school. This is how I was taught in college to do a dihybrid cross. Up until now, we've done monohybrid. Keep in mind, hybrid means blending or not, wrong choice of words. Not blending, but a mixture of gametes from mom and dad. Mono means one, di means two. So now we're looking at two traits at the same time and asking what are the chances of all the possible combinations of alleles from those two individuals. That means you have to have 16 squares when you're all done. Now you can do it this way. It's not hard, at least in terms of listing all the different probabilities. All you're going to do is say, what are the odds of getting a big R with a little y, then that big R with that little y, then that big R with that one, and that one with that, and that with this, and you get all these. That part's easy. But when it comes down to counting how many of each kind you have, you guys can get lost. You overlook one, or you didn't actually write a capital letter when you should have. So this does lend itself to being a little more of a pain in the arse to calculate. I want to show you a much easier way to do the same thing, get the same results, and it's a lot less complicated. We're going to go back and we're going to do this monohybrid cross right here. So it's also a good time for you to figure out if you can actually do this one cross. So we're going to do a dihybrid cross of big R, big Y. So big R, homozygous dominant for each trait, mated with probably homozygous recessive for each trait. And you're going to be asked to list all the possible genotypes from that mating. Come on, get up there. You guys done? The easiest way to do this is you're going to do two separate monohybrid crosses. I'm going to take the R trait and do a cross with the R's. Then I do a separate cross with the Y trait. So if I'm going to do big R crossed with big R, make my little box, and then the other individual has two recessive traits. And the more you do this, the more automatic the answer becomes. Homozygous dominant or the homozygous recessive always produces heterozygous dominant offspring. There's no other thing you can get unless weird mutations happen. And that would be the same thing with the Ys, right? Homozygous dominant crossed with homozygous recessive. Let's make it a loopy Y. And if you were to look at this, you would know you'd get nothing but heterozygous individuals. And it just takes practice being able to recognize that. All right, but now what if we mate the offspring? What if we took an individual who was, well, let's just do this. So everybody's going to be big R, big Y, little y. And I want to mate them with another individual of that same genotype. Now it's going to get complicated, you would think. We do the R's like we did over here. So I'm going to do the R's first. Big R, little r. and then my big Y, little y. So I'm gonna set the Y's up first. Need a little more room. And then we're gonna fill them in. I'm gonna wait a moment to make sure you guys are actually able to do this. Because you'll have to do a monohybrid cross for sure on your exam. Make sure everybody gets this part down. Shouldn't take you long. All right, hopefully you will all get this. Did you all get that? Mm-hmm. All right. Now you might be asked to list all the genotypes. So now I want you to list all the possible genotypes for each separate trait. List all the genotypes for this monohybrid, and list all the genotypes for that monohybrid. And again, that shouldn't take you too long to do. All right, so what am I going to list for the R traits? What are my different genotypes? All right, so we got a big R, what else do we have?
Little r.
Big R, little r, and what else do we have?
A little r.
Okay, those of you that are not talking, are you saying the same thing in your minds? You have the same idea? Mm-hmm. Right, you have homozygous dominant, heterozygous dominant, and homozygous recessive. We have those, we have those, and we have those. So as I write them down, I just put a little check mark in them. That means I've accounted for them, so I don't have any empty boxes when I'm all done. How about the probability of getting each of those genotypes? So what is the probability of getting each of those genotypes?
1 out of 4 and 2 out of 4.
1 out of 4, 2 out of 4 for who?
2 out of 4 for the R, the big R and the little r, and then 1 out of 4 for the big R and the little r.
For the big R and the, well, when you say the big R and the little r,
Well, basically,
I kind of go right here.
Well, I was, yeah, that's 2 out of 4.
Right, but when you said 1/4 for the big R and the little r.
Oh, I was talking about like the double.
That's where I go.
I should say double big R. That's what I meant to say.
Double big R, okay.
That's what I meant to say.
So what is the probability of getting this one? Homozygous
1 out
dominant.
of 4.
Okay, heterozygous dominant.
2 out of 4.
Homozygous recessive.
1 out of 4.
Right, that's how you want to refer to them. Say homozygous dominant, right? Heterozygous dominant and homozygous dominant.
Could you give a percentage?
I mean homozygous recessive. Could
Recessive, yeah.
Give a percentage instead of?
Sure, 25, 50, 25. Yeah, that'll be fine. It might be easier if you do the fraction part first, especially when it comes time to doing part 2 of this. But I mean, if I were to do just a monohybrid, yeah, I'm fine with that completely. All right, so we can look at a 1/4 probability, right? A 1/2 probability, and a 1/4 probability. What are the genotypes for the Ys?
The same.
They're the same, right? Big Y, big Y. That's known as what? What vocabulary word can I use to describe those?
A dominant.
Homozygous dominant.
Yeah.
And the vocabulary word for this one? Heterozygous dominant, and then homozygous recessive. John, do that. There we
That
go.
word is so crazy.
And the probabilities are going to be the same.
Why do we call it heterozygous dominant when if it's heterozygous, then it's obviously dominant?
Yeah, it's just to sort of keep in line with the
Good question.
flow. Now, if you look at this, even in terms of ratios, right? I've got a 1 to 2 to 1 ratio, right? I've got 1 of these, 2 of these, 1 of those. These patterns become ingrained in your mind as you do simple genetics. You will always get those kind of ratios when you have a heterozygous dominant trait crossed with another heterozygous dominant trait. All right, so that's the easy part.
But remember, all you're really doing is you're calculating what are the odds of, let's say, a sperm cell having big R compared to big R, little r, or a sperm cell having the big Y, or the big Y, little y. But these are just two of the traits in an individual. So now we need to really ask, what are the odds of that sperm cell having a big R along with a big Y? Because when we have thousands of traits, so we need to now do a probability issue. So am I going to be asking, let me just draw my little big DNA torpedo here. If it's a big R with a big Y, are you asking yourself, what are the odds of that sperm having a big R, or a big Y?
Yeah.
Or are we saying, what are the odds of having a big R, and a big Y?
And?
So if you use the word and in statistics, probabilities,
Yeah.
are you going to be adding or multiplying?
Adding, no.
If you're going to use the word add, okay, we're going to use the word add, we've got it. If you're going to use the word and, we're going to be multiplying out, because it's this and that, right? So we need to sort of have a little accounting, bookkeeping method to do this. So the first thing you do is you do a monohybrid cross. Then you list all the genotypes from that monohybrid cross. Then you list all the probabilities. Now, just for a monohybrid, percentages are fine, right? It'll be easier if you do fractions, because now we have to multiply each independent event with the other.
Yeah.
So doing a 50% times 50% might get a little confusing. If you can do it, that's fine. So here's what I do. I start from the top of this one, and I'm simply asking myself, what are the odds of the sperm cell having big R, and a big Y? So I'm going to go over here, and then that would be listed as
16.
big R, big Y, and what is the answer to our question?
1/16.
You get 1/16, right? Because we're multiplying the odds of getting this and that. And when you and, you multiply. So that equals 1/16. Let's make sure everybody is okay with that one part. All right? So now we want to keep the same thing and say, all right, well, now I know the chance of big R with a big Y. What about the odds of having a big R with a big Y, little y? What if it binds with this allele instead, due to meiosis and crossover independent assortment? Well, all right, then I'm simply going to then do it this way. So now you've got big R times the probability of big Y, little y.
Which would be Y
And now what is that probability?
1/8.
You get 1/8, right? Because 1/4 times 1/2 is 1/8. Now, to keep things simple, since we already have a denominator in 16ths, let's convert 1/8 into 16ths. Oh my God!
It's
So what would that be?
Two.
That's 2/16ths. All right. Well, then we want to also ask, okay, fine, that's that. But what if it was a big R binding with a little y? Then we could say, all right, well, let's look at that probability instead. So then I'm going to get big R. And in a moment, you might begin to see a pattern that's going to show up. The little y. And of course, that answer is,
1/16.
we get 1/16. 1 to 2 to 1. Same thing as 1 to 2 to 1. All right, great. That means we have now taken account for all the different possible combinations of alleles involving big R.
Okay.
We now have to do the same thing for big R and little r.
Now,
It's a mixing game. So I say, all right, I've done that one. Now let's do big R with the other R. I'll change colors so you can simply just see what we're doing. We're going to take the probability of this one times the probability of that one. Probability of this one times the probability of that one. Probability of this one times the probability of that one. So I guess I could have done that in red, but we'll do this in blue. So this would then be big R, little r, big R, little r. I'm gonna do it 3 times. I got enough room. Big R, little r, with big Y, big Y, big Y, and little y, little y. Now 1/2 times 1/4 is 1/8, but 1/8 and 16ths equals 2/16.
What if you put 1/8?
Well, you're gonna need to keep everything simple and straight, so you're gonna have to be able to convert 8ths into 16ths to make sure you haven't screwed up.
I just might forget that's
So
all.
if you're in your head, you can realize that a 1/8 is 2/16, that's fine.
I just gotta remember to do
But
that.
it'll just be simpler this way, because we all have the same common denominator.
Yeah.
Then you have 1/2 times 1/2, which is what?
1/4.
And 1/4 in terms of 16ths would be 4/16. And then we have 1/2 times 1/4, which is then going to be 1/8, and 1/8 in 16ths is 2/16. So make sure I haven't lost anybody with this part. It's the same thing as we did for the big R's. We're simply saying I'm going to start here and look at the probability of each and every one. I've done that, then I go here and do the probability of each and every one with that one set of alleles. Alright, now we got to do the bottom one. Notice that I've got 3 from this one, 3 from that one. I'm going to get 3 from this one. So I can simply just write that down 3 times. Big R, or little r, little r, little r, little r, little r, little r. And then I simply just take this, copy it, and put it right there. That's what I'm basically doing. Keep the colors the same. So that would be then big Y, big Y, big Y, little y, little y, little y. And then we go 1/4 times 1/4 equals 1/16. 1/4 times 1/2, oops, is 1/8 into 16ths, makes it 2/16. And 1/4 times 1/4 is 1/16.
Yeah.
And again, it's a 1 to 2 to 1. A 1 to 2 to 1. A 1 to 2 to 1. And it's this nice little pattern you begin to see. But now you can see we have a total of 16. At least we should have 16 different possible gamete combinations we can look at overall in terms of mixing and matching. Some will be replicated, some will not. Now the best thing is, not the best thing, the other idea is when you look at probabilities, all probabilities must add up to 1.
Yeah.
So we got to make sure that we've actually accounted for everybody correctly. So all these fractions should equal 1, which is why it's always easier to do it as a common denominator. Because if they're all 16ths, then all I got to worry about is making sure that the numerator equals 16. So 1 and 2 are 3, 4, 5, 6. 6 and 4 are 10, 12, 13, 14, 15,
16.
thank God, 16. Didn't screw up. 16 over 16 equals
1.
That's how you know you have it correct. Now imagine doing a tri-hybrid cross. How would you do it?
Same thing, but triple. It's like,
No, you're right. You would do 3 separate monohybrids, and then it would be simply, if we just put one more over here, let's just make it big Q, little q, and then we'll do a big Q, little q. So Q, Q, and let's, I'm going to have, so big Q, big Q, big Q, little y. Stop that. So it's a 1 to 2, oops, to 1 ratio. I would simply have to do this. Big R times big Y times big Q equals my probability. Big R, big Y times heterozygous Qs. Big R, big Y, homozygous Qs. Big R, heterozygous Y with big Q, big R, heterozygous Y with heterozygous Q, big R, heterozygous Y with homozygous Q. We're gonna be here a while,
Yeah.
but that's how you can simply do it. Even easier, you tell a computer how to do it. Alright? But this is what I had to do in college before computers were really that accessible. But this is a much more manageable way than doing the big possible 16 square. Alright? You can very easily calculate all the different alleles and gametes, but to go through and make sure you've got all the ones that could possibly be, you know, big R, little r, big Y, big Y. Because what if I were to ask you that we had a mating between these two individuals? What are the odds of getting someone who is heterozygous for both traits? What are the odds of getting someone who is heterozygous for both traits? Well, these are homozygous, that won't work. Those are homozygous, those won't work. The only one's actually heterozygous for both is right there. That's easy to do. 4 out of 16. But if you do with a big square, you got to go through every little. Okay, there's one. There's two. How many do I have? I forgot already. Got to start all over again. Right? So this makes it a much easier way of doing it.
Mm-hmm.
If I were to ask you, what are the odds of getting someone who's homozygous recessive for this? Then you simply go, who's totally homozygous recessive? Can't be those. Can't be those, because they each have a big R in them right off the bat. These are homozygous recessives, and then you're a homozygous with a dominant, there, 1/16. It makes it really easy to look at whatever I ask you guys to tell me the chances of something showing up. That last little bit that you get, right? If I were to ask you what are the odds of getting someone who is heterozygous for the R's and homozygous for the Y's? Heterozygous for the R's, homozygous for the Y's. He wants heterozygous for the R's, so it can't be those. Those are homozygous. These are homozygous for the R's, so I have to focus in here. He wants heterozygous for the R's and homozygous for the Y's. There it is. 1/8. 2 out of 16. You guys okay with that?
Okay.
And those of you that are just staring at me, you need to say, what the hell are you just doing? Or, I've got it.
I guess I
You only said homozygous. You didn't say homozygous dominant or recessive. So if you just say heterozygous R as a homozygous, would it be 4/16? State dominant or recessive?
Perhaps yes, yeah. So if I were to, ask you what are the odds of getting someone who's heterozygous for the R's, then you can simply just do all of these. If I say what are the odds of getting someone who's homozygous for the Y's, it would be that, and that, be that one, and this one, and that one, to go through them. Okay. Anybody? Yeah.
The baby will be 50%, so in here.
The baby will be 50.
Yeah, the offspring will be 50%.
50% what?
Like how many percent have to be to make a baby? How many percent does it have to be?
Well, you need all the genes from mom and all the genes from dad. I'm not sure I understand what you're asking.
Okay.
So, what you're looking at is, if this were the chances of sperm cells having these genotypes, right?
Yeah.
Or you could also ask, what are the odds of a sperm cell having, so a sperm cell having this genotype, right? So it's big R, little r, right, with a big Y, little y. And then you would ask, well, what are the odds of a mom's egg having this genotype? So then be little r, big Y, big Y. So now you know the odds of an egg would be 1/16 chance of having those alleles. You know the sperm would be 4/16 having those alleles, right? All right, then you would have to then cross and match them. You'd have to breed them and do this cross between